3D Effect Mesothelioma. 3D models of mesothelioma in the study of mechanisms of cell survival. Abstract. Three-dimensional models can provide another experimental system for investigating many aspects of tumor biology, particularly of cell death and survival. These models, both in vitro and ex vivo, may be ideally suited for studying mesothelioma. Here we discuss the reports that are currently available on this subject and highlight areas in which 3D biology may provide insights into the mechanisms of cell death and survival in mesothelioma. It has long been known that single cells or monolayers are more sensitive to cell death than 3D aggregates or spheroids, which acquire resistance called multicellular resistance. Possible survival mechanisms that have been studied using multicellular spheroids, in vitro, and tumor fragment spheroids, ex vivo, include growth factor pathways, calcium-dependent cell-cell adhesion, resistance to drug penetrance, gene expression, epigenetics, BCL2 anti-apoptotic family expression, and autophagy. In general, cells in a 3D setting show many differences compared to the same cells in a 2D monolayer. Many of these differences appear to contribute to cell survival. In many cases, these features can be shown to be more similar to those in the actual tumor. Thus, whereas 3D studies offer more challenges for study, they offer the opportunity for new insights not possible in 2D studies. They may thus offer an intermediate model between the monolayer and in vivo studies and hopefully lead to discoveries that can be tested in clinical trials to benefit patients. In vitro characterization of cisplatin and penetrance effects in malignant pleural mesothelioma 3D culture phenotypes. Abstract. Malignant pleural mesothelioma, MPM, is an aggressive cancer with poor prognosis. The main treatment for MPM is doublet chemotherapy with cisplatin and pemetrexed, while ongoing trials test the efficacy of pemetrexed monotherapy. However, there is lack of evidence regarding the effects of cisplatin and pemetrexed on MPM cell phenotypes, especially in three-dimensional, 3D, cell cultures. In this study, we evaluated the effects cisplatin and pemetrexed on cell viability using homologous cell-derived extracellular matrix, HECM, as substratum and subsequently in the following 3D cell culture phenotypes: tumor spheroid formation, tumor spheroid invasion, and collagen gel contraction. We used benign mesothelial MET5A cells as controls and the MPM cell lines M14K, epitheliid, Msdo, phasic, and ZL34, sarcomatoid. Cell viability of all cell lines was significantly decreased with all treatments. Mean tumor spheroid perimeter was reduced after treatment with pemetrexed or the doublet therapy in all cell lines, while cisplatin reduced the mean spheroid perimeter of MET5A and Msdo cells. Doublet treatment reduced the invasive capacity of spheroids of cell lines into collagenous matrices, while cisplatin lowered the invasion of the Msdo and ZL34 cell lines, and pemetrexed lowered the invasion of MET5A and ZL34 cell lines. Treatment with pemetrexed or the combination significantly reduced the collagen gel contraction of all cell lines, while cisplatin treatment affected only the MET5A and M14K cells. The results of the current study can be used as an in vitro 3D platform for testing novel drugs against MPM for ameliorating the effects of first-line chemotherapeutics. Keywords: 3D cultures, cisplatin, homologous cell-derived extracellular matrix, malignant pleural mesothelioma, mesothelial cells, pemetrexed, Plural, tumor spheroid. Introduction. Malignant pleural mesothelioma, MPM, is a rare and fatal cancer causatively linked to asbestos exposure that arises from the pleural mesothelium. MPM cases comprise 80% of the total mesothelioma cases that may also occur in the peritoneum and the pericardium. Despite asbestos being a regulated material in many countries, there are several other similar, fibrous materials that have been linked to MPM development. These materials could potentially prolong the asbestos-induced MPM epidemic, which is expected to reach its peak in around 2020. The median survival of MPM patients is 12 months following diagnosis, and the treatment options, chemotherapy, radiotherapy, surgery and palliative therapy, depend on the stage of the disease. Since MPM is asymptomatic at early stages, most patients are diagnosed at stage 3, and thus cytoreductive surgery is usually not applicable. Chemotherapy comprises the main therapy option for MPM patients or is used as a follow-up therapy after surgery. The frontline chemotherapy is a combination of cisplatin, cisdiaminidichloroplatinium, Cpdd, and pemetrexed, disodium salt of a synthetic pyrimidine-based antifolate, PAM, at doses of 75 mg per square meter and 500 mg per square meter, respectively, since it has been shown to confer a longer survival advantage. Cpdd cross-links with the DNA purine bases, interferes with DNA repair mechanisms, and causes DNA damage, thus leading to cell apoptosis. PAM inhibits 3 enzymes critical for the purine and pyrimidine synthesis and the folate metabolism. MPM severity influences treatment response and depends on the histological subtype, sarcomatoid, phasic, and epithelioid MPM, with sarcomatoid MPM being the most aggressive and epithelioid MPM being the least aggressive. Plural mesothelial cells undergo epithelial mesenchymal transition, EMT, during MPM development. EMT is a process that renders epithelial cells with mesenchymal properties during cancer development and leads to more aggressive phenotypes. The components of the extracellular matrix, ECM, that are produced by cells are critical for EMT and tumor metastasis. Moreover, a modified ECM composition due to malignant cells can induce favorable characteristics in cancer cells such as chemoresistance. In physiological conditions, ECM is critical for cell homeostasis, providing the proper three-dimensional, 3D, tissue architecture wherein the cells can thrive. Therefore, the ECM component should be taken into account in the in vitro study of cell phenotypes pertinent to tumor development and progression. Furthermore, cells cultured in 3D exhibit a different gene expression pattern of genes involved in cell signaling, proliferation, adhesion and apoptosis pathways as compared to monolayer, 2D, cell cultures. In line with the above, investigations of cancer cell phenotypes in the context of administered chemotherapeutics would yield the most clinically relevant results when assessed in 3D cultures with the use of HECM. In terms of MPM and Cpdd, PAM doublet treatment, there is a scarcity of data that demonstrate the effects of the treatment on 3D cultures. In order to fill this gap in the literature, we investigated the effects of Cpdd, PAM and Cpdd, PAM doublet treatment on the ability of MPM cells to form tumor spheroids, on the invasiveness of these tumor spheroids and on the property of the MPM cells to contract in a 3D collagen gel environment. Results: cell viability was significantly decreased after all treatments in all cell types on homologous cell-derived ECM substratum. Cells were subject to Cpdd, PAM or Cpdd plus PAM treatments in order to assess their survival. 10% FBS-RPMI was used as control, con. Cell viability with treatments was compared to control that was arbitrarily set as 100%. In MET5A cells, the mean cell viability was significantly lower with all drug treatments, and Cpdd plus PAM significantly reduced the cell viability compared to PAM or Cpdd. Con. 100.0 plus minus 2.75%, Cpdd, 68.54 plus minus 8.80%, PAM, 63.64 plus minus 1.39%, Cpdd plus PAM, 44.21 plus minus 1.42%, p less than 0.001. In M14K cells, the cell viability was significantly reduced by all drug treatments. while PEM-treated groups had significantly reduced cell viability compared to CpDD alone. Con 100.0 plus minus 4.42%, CpDD 63.83 plus minus 1.45%, PEM 21.84 plus minus 3.34%, CpDD plus PEM 21.02 plus minus 1.79%, p less than 0.001. In Msdo cells, the mean cell viability was significantly reduced by all drug treatments. Again, PEM-treated groups had significantly reduced cell viability compared to CpDD. Con 100.0 plus minus 2.14%, CpDD 74.99 plus minus 3.48%, PEM 42.13 plus minus 1.50%, CpDD plus PEM 43.14 plus minus 1.39%, p less than 0.001. Finally, in ZL34 cells the viability was significantly lowered with all drug treatments, and also in this case, PEM-treated groups had significantly reduced cell viability compared to CpDD. Con 100.0 plus minus 2.26%, CpDD 76.42 plus minus 1.69%, PEM 41.43 plus minus 0.92%, CpDD plus PEM 36.86 plus minus 1.15%, p less than 0.001. Mean values of cell viability plus minus some without or with treatments, CpDD, PEM, or CpDD plus PEM, expressed as percent of controls, cells with 10% FBS RPMI. A, MET5A cells, N equals 13, B, M14K cells, N equals 16, C, Msdo cells, N equals 16, D, ZL34, N equals 15 or 16. P less than 0.001 versus controls, at p less than 0.01 and at p less than 0.001 versus CpDD, hash p less than 0.05 versus PEM. Tumor spheroid formation in all cell types was significantly reduced with CpDD and PEM combined treatment. Spheroids were formed using the hanging drop method in culture media containing drugs, and comparisons were made regarding spheroid perimeter in pixels. The mean perimeter of the spheroids formed using culture media without drugs was arbitrarily set as 100% for each cell line. In MET5A cells, the mean spheroid perimeter was significantly reduced with all drug treatments, while CpDD plus PEM treatment significantly reduced the mean spheroid perimeter compared to CpDD alone but not PEM alone. Con 100.0 plus minus 2.62%, CpDD 86.63 plus minus 1.73%, PEM 82.86 plus minus 2.15%, CpDD plus PEM 74.23 plus minus 2.01%, p less than 0.001. In M14K cells, the mean spheroid perimeter was significantly reduced in the PEM-treated groups, con 98.44 plus minus 2.56%, CpDD 104.0 plus minus 3.57%, PEM 74.25 plus minus 2.70%, CpDD plus PEM 78.87 plus minus 2.33%, p less than 0.001. In Msdo cells, the mean spheroid perimeter was significantly reduced with all drug treatments, while PEM-treated groups had reduced mean spheroid perimeter significantly more compared to CpDD. Con 100 plus minus 2.88%, CpDD 84.05 plus minus 1.39%, PEM 66.22 plus minus 1.49%, Cpdd plus PEM, 64.69 plus minus 1.13%, p less than 0.001. Finally, in ZL34 cells as in M14K cells, the mean spheroid perimeter was significantly reduced in the PEM-treated groups, con 100 plus minus 1.81%, Cpdd 97.55 plus minus 1.04%, PEM 77.58 plus minus 0.99%, Cpdd plus PEM 78.10 plus minus 1.87%, p less than 0.001. Mean values of spheroid perimeter plus minus some without or with treatments, Cpdd, PEM, or Cpdd plus PEM, expressed as percent of controls, cells with 10% FBS RPMI, along with representative microscopy images from each group. A. MET5A cells, N equals 13 to 17, B. M14K cells, N equals 10 to 15. C. Msdo cells, N equals 15 to 18, D. ZL34, N equals 13 to 15. P less than 0.001 versus controls, at p less than 0.001 versus Cpdd. E. Images of spheroids of cell types arranged by rows and their corresponding treatments in a column-wise manner. Scale bars represent a length of 100 pixels. Cpdd and PEM combination significantly reduced the invasion of MPM spheroids in collagen gel matrix. MPM spheroids were embedded in collagen to evaluate the effects of Cpdd and PEM during invasion inside collagen gel matrices. The mean perimeter of the spheroids inside the collagen with 10% FBS RPMI was arbitrarily set as 100% for each untreated cell line individually. The MET5A cell spheroid invasion inside the collagen matrix was significantly lower in the PEM-treated groups, con 100.0 plus minus 3.01%, Cpdd 92.27 plus minus 2.20%, PEM 84.51 plus minus 1.72%, Cpdd plus PEM 86.98 plus minus 2.19%, p less than 0.001. The M14K invasion inside the collagen matrix was significantly reduced when treated with Cpdd plus PEM, and Cpdd plus PEM significantly reduced the invasion compared to PEM. Con 100.0 plus minus 9.76%, Cpdd 75.93 plus minus 4.40%, PEM 95.3 plus minus 6.63%, Cpdd plus PEM 64.61 plus minus 5.41%, p less than 0.001. The Msdo cells invaded significantly less when treated with Cpdd or Cpdd plus PEM. Con 100.0 plus minus 9.79%, Cpdd 68.20 plus minus 4.47%, PEM 77.28 plus minus 4.60%, Cpdd plus PEM 61.16 plus minus 5.00%, p less than 0.001. Finally, the ZL34 cells invaded significantly less with all drug treatments, con 100.0 plus minus 2.36%, Cpdd 74.87 plus minus 4.78%, PEM 65.32 plus minus 4.77%, Cpdd plus PEM 62.25 plus minus 4.10%, p less than 0.001. Mean values of perimeter of spheroid invasion plus minus some without or with treatments, Cpdd, PEM, or Cpdd plus PEM, expressed as percent of controls, cells with 10% FBS RPMI, along with representative microscopy images from each group. A. MET5A cells, N equals 16 to 21. B. M14K cells, N equals 19 to 22. C. Msdo cells, N equals 13 to 19. D. ZL34, N equals 15 to 23. P less than 0.01 and P less than 0.001 versus controls, P less than 0.01 versus PUM. E. Images of spheroids invading into the surrounding collagen matrix, arranged by rows, by cell type, and their corresponding treatments in a column-wise manner. Scale bars represent a length of 100 pixels. PUM alone and Cpdd in PUM combination significantly reduced the contraction of collagen gels. Epithelial mesenchymal transition, EMT, in 3D is a phenotype that can be studied with cells embedded in collagen gels. Cells progressively organize and contract the collagen gel post-polymerization within 30 minutes. We performed 3D EMT studies without the drugs, and the final gel areas were evaluated as a measure of 3D EMT. Gel contraction with drugs was compared to cells treated with 10% FBS RPMI, 100%. Regarding MET5A and M14K, the mean gel area was significantly higher with all treatments. MET5A, con, 100.0 plus minus 0.94%, Cpdd, 116.1 plus minus 3.54%, PUM, 119.8 plus minus 3.64%, Cpdd plus PUM, 123.0 plus minus 3.28%, P less than 0.001, M14K, con, 100.0 plus minus 2.31%, Cpdd, 127.9 plus minus 2.08%, PUM, 157.9 plus minus 11.73%, Cpdd plus PUM, 160.3 plus minus 8.63%, P less than 0.001. In Msdo cell line, the mean gel area was significantly increased when treated with PUM or Cpdd plus PUM. Compared to Cpdd, both PUM and Cpdd plus PUM significantly reduced the contraction of Msdo cells, while Cpdd plus PUM was more potent than PUM alone. Con, 100.0 plus minus 1.03%, Cpdd, 104.4 plus minus 1.76%, PUM, 130.8 plus minus 2.29%, Cpdd plus PUM, 140.2 plus minus 2.66%, P less than 0.001. In ZL34 cell line, the mean gel area was significantly higher in the PUM-treated groups compared to control or Cpdd alone. Con, 100.0 plus minus 1.14%, Cpdd, 103.2 plus minus 1.56%, PUM, 130.8 plus minus 1.93%, Cpdd plus PUM, 130.1 plus minus 3.39%, P less than 0.001. Mean values of gel area plus minus some without or with treatments, Cpdd, PUM, or Cpdd plus PUM, expressed as percent of controls, cells with 10% FBS RPMI, along with representative microscopy images from each group. A. MET5A cells, N equals 12, B. M14K cells, N equals 12, C. Msdo cells, N equals 16, D. ZL34, N equals 11 to 12. P less than 0.01 and P less than 0.001 versus controls, at P less than 0.01 and at P less than 0.001 versus Cppd, P less than 0.01 versus PUM. E. Images of contracted gels with cell types arranged by rows and their corresponding treatments in a column-wise manner. Discussion. In this paper, we opted to create a well-characterized in vitro 3D platform for assessing the efficacy of the current chemotherapeutics used for the treatment of MPM that can be further used in order to assess adjuvant treatments. Under the scope of the above, we evaluated the effects of Cpdd in PEM in the context of MPM cell viability, tumor spheroid formation, tumor spheroid invasion, and collagen gel contraction in MET5A, M14K, Msdo, and ZL34 cell lines. Treatment with Cpdd, PEM or Cpdd plus PEM reduced the cell viability of all cell types as expected, but PEM or Cpdd plus PEM was more potent at reducing MPM cell viability as compared to Cpdd treatment alone. The cytotoxic effects of Cpdd and PEM on Msdo cells have been previously shown, demonstrating lower cell viability. Cpdd has been reported to lower the viability of MET5A and Msdo cells at a dose of 20 meters over 24 hours on polyol-lysine-treated plates. In our experiments, we used 10 meters of Cpdd over 48 hours on cells deposited on homologous cell-derived ECM. ECM derived from invasive cells induces 5-fluorouracil, 5-FU, resistance in height 29 colorectal cancer cells when compared to height 29 cells that were deposited on cell-derived ECM either from non-invasive or from normal colorectal cells. Moreover, based on the origin of cell-derived ECM, cells exhibit different gene expression levels of ABCB1 and ABCC1 that are involved in efflux of 5-FU. Thus, the use of homologous cell-derived ECM for cell viability assays of malignant cells is a better representation of the in vivo tumor ECM architecture as it entails potential involvement of drug resistance. Our results show that, in 2D flat surfaces, PEM is the main driver of cytotoxicity in malignant cell lines, while in benign mesothelial cells, there is a synergistic effect of Cpdd and PEM. During 3D growth, 3D invasion, and 3D MT, our data shows that Cpdd monotreatment demonstrated context-dependent inhibition, while in most cases Cpdd plus PEM was not superior to PEM alone. With respect to spheroid formation, all treatments reduced the perimeter of MET5A and Msdo spheroids, while M14K and ZL34 spheroids were smaller only when treated with PEM or the combination of the drugs. The importance of tumor spheroids in MPM studies has been highlighted before. Spheroids have been shown to differentially express genes as compared to monolayers, so the use of 3D cultures demonstrates more accurately the response to drug treatments, mimicking the conditions of the human body to a better extent. Through the application of the in vitro spheroid formation assay, the phenotypes of cell adhesion and cell proliferation are concomitantly assessed in an arrangement that resembles the spheroid found in pleural fluids of patients with MPM. We have shown that spheroids from all cell lines were affected by PEM treatments, PEM or Cpdd plus PEM, while Cpdd did not reduce the perimeter of the spheroids of M14K and ZL34 cells. This suggests that the process of spheroid formation of M14K and ZL34 cells is resistant to Cpdd treatment. This finding is in agreement with a previous study showing that populations of H28, H2052, sarcomatoid MPM, and ACC meso4, epitheliid MPM, cells that were expressing high concentrations of aldehyde dehydrogenase, ALDH, and were CD44 positive developed spheroids despite of Cpdd treatment. Based on the above, Cpdd seems to affect spheroid formation in a histotype-dependent manner, while PEM was able to pose its effects regardless of the cell type, rendering PEM the critical factor for reduction of the spheroid formation in MPM patients. Furthermore, we have recently published that adding 2-deoxyglucose, 2-DG, to doublet treatment Cpdd plus PEM had additive effects in terms of proliferation inhibition only in 2D cultures and not in 3D spheroids in Msdo and ZL34 cells lines, which is suggestive of a significant challenge posed in the treatment of 3D tissue structures. On the other hand, a recent study using the FAK inhibitor BI853521 showed that MPM3D spheroid growth was lowered by FAK inhibition, while this was not the case in 2D cultures. Collectively, these data point to the importance of the adoption of 3D cultures in MPM treatment testing, and recently, the use of patient-derived 3D organoid cultures for individual drug testing has been published. Tumor spheroid invasion is a 3D assay that demonstrates the invasion potential of tumor spheroids within an collagenous matrix. Cpdd reduced the invasion of Msdo and ZL34 cells, PIM affected MET5A and ZL34 cells, while the combined treatment significantly reduced the spheroid invasion of spheroids from all cell lines. Collagen drop drug sensitivity test, CDDST, is a method evaluating the effectiveness of a drug treatment on biopsies of malignant tissues focusing on their invasiveness. CDDST with Cpdd has been reported, but the effects of combination of Cpdd and PIM on different MPM histotypes are unknown. Therefore, the methodology employed in the current paper demonstrates that, in the scope of the limited availability of patient biopsies, a collagen tumor spheroid invasion assay using chemotherapeutics would be a valuable tool to assess patient-specific drug effectiveness in the clinical management of mesothelioma. To further substantiate this, 3 recent studies have also recently shown that perfenidone and erlotinib plus photodynamic therapy were able to inhibit the 3D growth of MPM spheroids in collagen igel and matrigel, respectively. Matrigel is a mouse sarcoma-derived ECM that may induce other pathways implicated in cell E.C.M. signaling due to its cancerous origin, but still is an important tool in such studies. However, using either commercially available collagen or in-house collagen, as was done in our study, provides a better representation of the benign nature of the surrounding ECM. Gel contraction is a 3D assay that assesses the mesenchymal potential of cancer cells by testing the contraction of malignant cells inside an ECM component, as is the case in tumor progression. The gel contraction assay is a measure of the degree of native mesenchymal transition of the cells, as measured from the area of the contracted gels. In our model, we used rat tail collagen gel areas resulting from contraction over a period of 24 hours. PIM decreased the contraction of cells embedded in collagen gels in all cell lines tested. Cpdd only decreased the contraction of MET5A and M14K cells, while the combined treatment of Cpdd and PIM decreased the gel contraction in all cell lines. It has been reported that mouse uterine mesothelial cells that have undergone EMT express alpha smooth muscle actin, alpha SMA, and develop a contractile phenotype typical of smooth muscle cells. Our cells demonstrated contractile characteristics that were inhibited with the use of chemotherapeutics. Materials and methods. Cell culture. The human cell lines MET5A, benign immortalized mesothelial cells, M14K, epitheliid MPM, Msdo, phasic MPM, and ZL34, sarcomatoid MPM, were used and were kindly provided by Professor Ioannis Kalamenides, National and Kapodistrian University of Athens, Athens, Greece. Cells were cultured with 10% fetal bovine serum RPMI, FO804, sigma, Street Louis, Missouri, USA, 2L-glutamine, G7513, sigma, 1% penicillin, streptomycin, P4333, sigma, and 0.5% width, V-plasmocin, ANT-MPP, and vivo gen, Toulouse, France. In a 5% CO2 incubator, cells were synchronized by serum starvation, 0.5% FPS RPMI, over 24 hours prior to all experiments. Drug treatments. Filter sterilized 10-5M cisplatin, Cpdd, PHR 1624, sigma, and 2 times 10-4M pemetrexed, PIM, CDSO24404, sigma, and their combination, Cpdd plus PIM. were used. Acid-soluble rat tail collagen preparation. Tails were obtained from euthanized white Norwegian Sprugdorley rats that were designated for disposal, kindly provided by Dr. Konstantinos Dimas, Faculty of Medicine, University of Thessaly, Larissa, Greece. The tails were cut along the anterior-posterior axis at regular intervals without penetrating the core of the tail. Small 1 cm portions of intact tail were pulled apart until cords of white material in between were stretched. Such strings of collagen were collected, weighed and immersed in 95% ethanol and then air-dried in a laminar flow hood. The dried collagen cords were subject to leaching with sterile 0.2 m acetic acid, in a sterile conical flask on a magnetic stir plate for 16 hours at 4 degrees Celsius. The solution was then subject to clarification by centrifuging at 2,500 grams for 20 minutes, 2 times in order to sediment undissolved protein. The resulting collagenous material was then stored in sterilized glass bottles at 4 degrees Celsius. Cell viability assay. Cells, 5 times 104 cells per milliliter, were seeded in 0.5% FPS RPMI media for 24 hours in 96 well plates pre-treated with homologous cell-derived ECM as described previously. Afterward, cells were incubated for 48 hours with 10% FPS RPMI with the appropriate drugs. At the end of the assay, cells were washed with PBS, followed by fixation with 4% paraformaldehyde, PFA, then stained with 0.5% crystal violet and subsequently washed in running tap water. Plates were stained with 10% acetic acid after drying overnight. The acetic acid dissolution was then subjected to OD measurements at 570 nanometers. Each experiment had a replicate of 6 to 8 and was repeated 2 times. Tumor spheroid formation assay. 50 synchronized cells in 25 liters of media were used to form spheroids in suspension in a hanging drop format over 48 hours. In some suspensions Cpdd, PAM, or Cpdd plus PAM were included in the cell medium. Spheroids were imaged, and their perimeters were measured with image-edge software using the polygon lasso tool. Each experiment had a replicate of 5 to 9 and was repeated 2 times. Tumor spheroid invasion assay. Spheroids were formed as described above, and then pH-neutral collagen mix with 10% FPS RPMI was overlaid on top of the spheroids. Some of the collagen RPMI mixtures contained Cpdd, PAM, or Cpdd plus PAM and cultured for 48 hours. Spheroids inside the collagen were imaged, and their perimeters were measured using image-edge software with the polygon lasso tool. Each experiment had a replicate of 7 to 12 and was repeated 2 times. Gel contraction assay. All reagents were kept on ice. Neutralized collagen, 1.2 to 1.5 mg per milliliter, with 0.5 Mno and 1 times 106 synchronized cells per milliliter was used. The cells were embedded in 0.6 milliliters volume in 24 well plates. In some of the mixture, drugs were included at 2 times concentration. The gels were allowed to polymerize over 30 minutes followed by addition of 0.6 milliliters 10% FPS RPMI. The gels were then allowed to contract over 24 hours. At the end of the assay the plates were scanned, and the area of each gel was measured using image-edge software with the circular lasso tool. Each experiment had a replicate of 5 to 8 and was repeated 2 times. Statistical analysis. Analyses were performed using PRISM 7 for Mac, San Diego, California, USA. Normality of data was assessed by the D'Agostino and Pearson normality test. Data comparisons were performed with one-way ANOVA for parametric data or Kruskal-Wallis test for non-parametric data. All data are presented as mean plus minus sim. Values of p less than 0.05 were deemed as significant. Conclusions. In conclusion, our study evaluated the effects of Cpdd in PAM, the first-line chemotherapeutics, on different MPM cell lines representing the 3 different mesothelioma histological subtypes in 3D culture assays. The advantage of a 3D cell culture platform is that it can be used to assess the efficacy of adjuvant drugs in the context of MPM treatment.